Here's a question. Using genetics, can we find the origin of the Romani people? The Romani settled in Europe over 1,000 years ago. Using population genetics, I will show where from the South Asian subcontinent their ancestors came from, and, over time, how much European admixture has occurred within the Romani people. This is going to be a fun one for the anthropologists out there. I'm Dr. Krutik Patel, PhD, and welcome to this week's Scientific Journal Club. Firstly, we must define the Romani people as an anthropological population. This is a map I created using Minority Right Group International data. Here we show the estimated number of Romani within Europe. The largest total population exists in Turkey with more than 2.5 million and Bulgaria with more than 1.5 million. For the purposes of this anthropological essay, we will group these 10 to 12 million people as a single homologous population. Here are some caveats to our definition. We had no method of differentiating the Sinti people, a large subpopulation or clan within the Romani, so we had to include them. I do apologize though if this is culturally inaccurate. There are also the Dom people of Egypt, a sister population to the Romani. We are also not including them in our definition of the Romani population. Now that we have our definition sorted, let's discuss why the Romani could be described as an anomalous population amongst Europeans. They hold cultural, musical, and linguistic practices which have held since their descent from South Asia. Their descent from the Indian subcontinent has a range of evidence including cultural practices such as marriage customs and a caste system. Also, linguistic evidence has shown their language has been inherited from parts of Kashmiri, Punjabi, or Hindi dialects. While a linguistic and cultural heritage provides some semblance into the anthropological past of the Romani, the application of modern population genetics sheds much more light into answering the question, where specifically from the South Asian subcontinent the ancestors of the Romani arose from. Melegh et al. Performed a large single nucleotide polymorphism study to genotype a representative population of Romani people and contrast this against populations found in 51 unique geographic locations within the South Asian continent. We previously described what SNPs were in the cat video. Please click the pop-up above to find out more. This dataset included 158 samples making up the Romani population, 388 samples making up the South Asian population, and 1,456 samples making up the European population. Let's focus on the contrast between the DNA between the South Asians and the Romani people for now. Here, 53,928 shared SNPs were contrasted between the Romani population and 51 unique geographical populations from the South Asian dataset. The 51 geographical locations aggregated to 9 broader geographic locations. These were North Indian, Northwestern Indian, Northeastern Indian, Central Indian, South Indian, Southwestern Indian, Southeastern Indian, Pakistani, and a population representing the Andaman and Nicobar Islands. To understand the analysis performed in this study, we must first grasp the concept of genetic drift. That is, as more time passes, genes have more opportunity to mutate and diverge. Let's use a small segment of DNA to showcase this. From our starter population to generation 2, we see a G change to a C. Then in generation 3, we see an additional mutation as an A changes to a T. A new mutation arises in generation 4 and another in generation 5. Notice that these mutations are cumulative and earlier mutations seen in previous generations are inherited by later generations. Along comes our SNP array, which was designed to extract nucleotides 4, 6, and 9 from this segment of DNA. In this example, This leaves three nucleotides to determine the relatedness between the generations. And let's replace generation to population. And the concept remains the same. We have these three nucleotides to determine the relatedness. We can rework this as a crude measure of genetic distance, and this gives us an indication of genetic drift. Now let's take this concept further and introduce inheritance by descent, or IBD. This was used to identify genetic distance between the Romani and the 9 South Asian populations introduced before. Don't worry, this formula is not as scary as it seems. We'll go through it slowly. It is a function to measure the SNPs from two populations. We use a bit of algebra to call these populations I and J. In our study, we will use I as the Romani and J as one of the South Asian populations, for example, the North Indians. We assess each individual per population. Lower I represents individuals from population I, and lower J represents individuals from population J. We also take into consideration the population sizes. N is the population size for population I. This was 158 in the study. M is the population size for population J. That would be the total number of North Indian samples this study had in this example. Now that we have our algebra out of the way, we can continue with the individual contrasts. So, SNPs from individual 1 from population I are contrasted against individual 1 from population J. Then, individual 2 from I is contrasted against individual 1 from population J. And then individual 3 from population I is contrasted against individual 1 from population J. And so on. All individuals from each population will be contrasted against each other, and then the sum from all the individual contrasts between populations I and J is calculated. This will be divided by the product of N and M, giving us an average IBD score for these two populations. A higher IBD scoring indicates, on average, more common SNPs between the two populations and a lower amount of genetic drift. We see the lowest amount of genetic drift between the Romani and northwestern Indian populations, and this was seconded by the Pakistani population. This was followed by the Central and South Indian populations. One interesting result was that the Romani and North Indian populations did not have a particularly impressive IBD score. This evidence reshapes our conceived notion of where the ancestors of the Romani people descended from. While the linguistic evidence has held the North Indian languages like Punjabi and Hindi as possible root languages for the Romani language, it seems the northwestern part of India holds the most prominent evidence of a genetic root of the ancestors of the Romani people. This study also provided IBD scoring between the Romani population and several broad European populations, which showed the Central and Eastern European populations having the highest IBD scores. Furthermore, the lowest IBD score of the European population tests was South European. The scoring suggests, apart from three South Asian populations, the Romani people are much closer to the European populations than the populations found in the South Asian continent. This highlights a very important fact. The modern Romani people are, for all instances, Europeans. While their genetic history is interesting as they're unique outliers for recent European anthropology, we should not discount that their population has had over 1,000 years to settle, interact, and evolve into a new people. We see this clearly in another paper on the same topic. The authors of this other study concluded only 17% of the Romani population's current genetic makeup are derived from South Asian descent, while 83% are contributory to Caucasian descent. Alas, going into this in more detail would require a whole other video. There we have it, folks, the anthropological history of the Romani people. This was a genuinely fascinating video to make, and I hope you enjoyed it. Please like and subscribe for more of these types of videos, and I hope to see you all in next week's Scientific Journal Club.